Hello viewers, welcome to my channel Online Biology Tutorials. You are a living archive of generations, traits passed down like coded whispers. Type generation if you are ready to decode inheritance. Picture 4 boxes on a page. Inside them, possibility. 3, 2, 1. Let's go with the reading of the chapter Principles of Inheritance and Variation. It is chapter number 4 of class 12th NCERT. I hope the screen is very much visible to you. Yes, it is. So this is from the unit 7, Genetics and Evolution, one of the biggest units with maximum weightage. I'll come for the explanation for this chapter as well, but otherwise also I have pre-recorded playlist already on my channel. In case you are interested, do check out. It is going to be a long chapter, will take much time in comparison to the other 3 chapters that we are already done with. So stay connected till end. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal? Or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents, or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offsprings. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Human knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possess desirable characters. For example, through artificial selection, domestication from ancestral wild cows, we have well-known Indian breeds, example Sahiwal cows in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of these phenomena. 4.1 Mendel's Law of Inheritance. It was during the mid-19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years, from 1856 to 1863, and proposed the laws of inheritance in different organisms. During Mendel's investigations into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size, which gave greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing traits. Example, tall or dwarf plants, yellow or green seeded. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination cross pollination experiments using several true breeding bee lines. A true breeding line is one that, having undergone continuous self pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with the contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated, full or constricted green or yellow pods, and tall or dwarf plants. Table 4.1 is important. It is giving you an idea of all the 7 characters used by Mendel, studied by Mendel in the pea plant. These characters were stem height, flower color, flower position, pod shape, pod color, seed shape, and seed color. We will be studying in one shot what are these about. 4.2 Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel where he crossed tall and dwarf pea plants to study the inheritance of one gene. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. The generation is also called the filial progeny or the F1. Mendel observed that all the F1 progeny plants were tall, like one of its parents. None were dwarf. He made similar observations for the other pairs of traits. He found that the F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the filial 2 generation, some of the offspring were dwarf as well. The character that was not seen in the F1 generation was now expressed. The proportion of the plants that were dwarf were 1 by 4th of the F2 plants, while 3 by 4th of the F2 plants were tall. The tall and dwarf traits were identical to their parent type and did not show any blending. And that is all the offspring were either tall or dwarf, none were of in between height. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 generation, both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 generation. Based on these observations, Mendel proposed that something was being stably passed down, unchanged from parent to offspring through the gametes over successive generations. He called these things as factors. Now we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. They are slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character for height, capital T is used for the tall trait and small t for the dwarf. And capital T and small t are alleles for each other. Hence, in plants, the pair of alleles for height would be capital T, capital T, capital T, small t, and small t. Mendel also proposed that in a true breeding, tall or dwarf pea variety, the allelic pair of genes for height are identical or homologous, capital T, and small t, respectively. Capital T, and small t are called the genotype of the plant, while the descriptive terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype capital T small t? As Mendel found the phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other and hence is called the dominant factor while the other factor is recessive. In this case, capital T is dominant over small t for dwarfness, that is recessive. He observed identical behavior of all the other characters, trait pairs that he studied. It is convenient and logical to use the capital and lowercase of an alphabetical symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and small d for dwarf because you will find it difficult to remember whether capital T and small d are alleles of the same gene, character or not. Alleles can be similar as in the case of homozygotes like capital T and small t or can be dissimilar as in the case of a heterozygote capital T small t. Since the capital T small t plant is heterozygous for genes controlling one character, that's height. It is a monohybrid and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other, and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is a 50% chance of a gamete containing either allele, as has been verified by the results of the crossings. In this way, the gametes of the tall capital T plants have the allele capital T, and the gametes of the dwarf small t plants have the allele small t. During fertilization, the two alleles capital T from one parent, say through the pollen, and small t from the other parent then through the egg are united to produce zygotes that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygotes, the F1 and F2 plants can be understood from the diagram called Punnett square, as shown in figure 4.4. It has been developed by a British geneticist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and the left columns. All possible combinations are present in boxes below in the squares, which generates a square output form. The Punnett square shows the parental tall capital T male and dwarf small t female plants. The gametes produced by them and the F1 capital T small t progeny, the F1 plants of genotype capital T small t are self-pollinated. The symbols female and male are used to denote the female and male pollen of the F1 generation respectively. The F1 plant of the genotype capital T small t, when self-pollinated, produces gametes of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grains of genotype capital T have a 50% chance to pollinate the eggs of the genotype capital T as well as of the genotype small t. Also, pollen grains of genotype small t have a 50% chance of pollinating the eggs of genotype capital T, as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be genotypes capital T, capital T, capital T, small t, or small t, small t. From the Punnett square, it is easily seen that 1 by 4th of the random fertilizations lead to capital T, 1 by 2 lead to capital T small t, and 1 by 4 to small t. Though the F1 have a genotype of capital T small t, but the phenotypic character seen is tall. At F2, 3 by 4th of the plants are tall, where some of them are capital T, while others are capital T small t. Externally, it is not possible to distinguish between the plants and the genotypes capital T and small t and capital T. Hence, within the genotype pair capital T small t, only one character capital T tall is expressed. Hence, the character capital T or tall is said to dominate over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall. And in F2, 3 by 4th of the plants are tall, whereas 1 by 4th are recessive. This leads to the phenotypic ratio of 3 by 4th, wherein 1 by 4th are capital T, 1 by 2 are capital T small t, and 1 by 4th are small t, giving you a ratio of 3:1. But a genotypic ratio of 1:2:1. The 1 by 4 and 1 by 2 and 1 by 4 ratio of capital T, small t and small t is mathematical condensable to the form of binomial expression ax plus by whole square 2 that has the gametes bearing genes capital T or small t in equal frequency of 1 by 2. The expression can be expanded in this form, and this should definitely be seen from NCERT. Mendel self-pollinated the F2 plants and found that the dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations as well. He concluded that the genotype of the dwarfs was homozygous small t. What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraphs, it is clear that though the genotype ratios can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotype composition. That is, for example, whether a tall plant from the F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a test cross. In a typical test cross, an organism, pea plants here showing a dominant phenotype and whose genotype to be determined. He crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. Figure 4.5 shows the results of typical test cross where violet color flower capital V is dominant over white color flower small v. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? Using the genotypes of this cross, can you give a general definition of a test cross? Based on his observations on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called principles of laws of inheritance, the first law of dominance and the second law of segregation. 4.2.1 Law of Dominance. 1. Characters are controlled by discrete unit called factors. 2. Factors occur in pairs. 3. In a dissimilar pair of factors, one member of the pair dominates dominant and the other is recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3 to 1 obtained in the F2. 4.2.2 The law of segregation, which was the second law given by Mendel, universally applicable. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen in the F1 stage. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. So till now, whatever keywords you have imbibed through this reading, go ahead and write it in chat. We will start with 4.2.2.1 Incomplete Dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that is not resembling either of the two parents and was in between the two. The inheritance of flower color in the dog flower, snapdragon or antirrhinum species is a good example to understand incomplete dominance. In a cross between two breeding red flowered R, and a true breeding white flowered plant r, the F1 R r was pink. When the F1 was self-pollinated, the F2 resulted in the following ratio. R1, that was red. R that was 2, that were 2 in number, color was pink. One was white with the genotype r. Here the genotypic ratios were exactly as we would expect in any Mendelian monohybrid cross. But the phenotypic ratios had changed from 3:1 dominant is to recessive ratio. What happened was that the R was not completely dominant over r, and this made it possible to distinguish between R, small r as pink from R red and small r white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is a pair of alleles. Now, these two alleles need not always be identical as in a heterozygote. One of them may be different due to some changes, that is, it has undergone, about which you will read further on and in the next chapter, which modifies the information that particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume, as in more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for the production of the normal or less efficient enzyme, a non-functional enzyme, or no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotype or trait, that is, result in the transformation of a substrate S. Such equivalent allele pairs are very common, and if the allele produces a non functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will not be dependent on the functioning of the unmodified allele. The unmodified functioning allele will represent the original phenotype is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Let's go ahead and move to the topic 4.2.2.2, codominance. Till now, we were discussing crosses where the F1 resembled either of the two parents, dominance, or was in between, incomplete dominance. But in the case of codominance, the F1 generation resembles both the parents. A good example is different types of RBCs or red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by a gene. The gene I has three alleles, IA, IB, and small I. The alleles capital IA and IB produce a slightly different form of sugar, while allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three capital I gene alleles. It could be IA, IB are completely dominant over small I. In other words, when IA and small I are present only, IA will be expressed. Because small I does not produce any sugar, and when IB and small I are present, IB will be expressing itself because small I does not produce any sugar. But when IA and IB are present together, they both express their own types of sugars, and this is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are six different combinations of these three alleles that are possible, and therefore a total of six different genotypes of these three alleles that are possible, and therefore a total of six different genotypes of the human ABO blood group types are possible. How many phenotypes are possible? Please go ahead. This table is very important. See from NCERT that if a person is holding the blood group A, it could be homozygous or heterozygous. Same logic goes for B. It could be IB or IB small I. And if it is AB, it would be capital IA, IB, and if it is O, there will be two small I. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than two, that is, three alleles governing the same character. Since in an individual only two alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has two alleles, capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, capital B seeds are round and small b seeds are wrinkled. Heterozygous produce round seeds, and so capital B seems to be the dominant allele, but the starch grains produced are of intermediate size in capital B small b seeds. So, if starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. 4.3 Inheritance of two genes. Mendel also worked with and crossed pea plants that differed in two characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape and one that had seeds of green color and wrinkled shape. Mendel found that the seeds resulting from the crossing of the parent had yellow colored and round colored seeds. Here, can you tell which of the characters in the pairs yellow or green color and round and wrinkled were dominant? Thus, yellow color was dominant over green and round shape was dominant over the wrinkle. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols capital Y for dominant yellow seed color and small y for recessive green seed color. Capital R for round shaped seeds and small r for wrinkled seed shape. The genotype of the parents can then be written as capital R, capital Y, capital Y and small r, small y, small y. The cross between the two plants can be written down as shown in figure 4.7. Showing the phenotypes of the parent plants, the gametes capital R, capital Y and small y unite in fertilization to produce F1 hybrid as capital R, capital Y, small y. When Mendel self-hybridized the F1 plants, he found that 3 by 4th of the F2 plants had yellow seeds and 1 by 4 had green. The yellow and green color segregated in 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like the monohybrid cross. You must have done 4.7 numerous times as it is a dihybrid cross. Let's come on to 4.3.1 Law of Independent Assortment. In a dihybrid cross, the phenotypes round, yellow, wrinkled, yellow, round green and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green with 3 round, 1 wrinkled. This derivation can be written as follows. So 9 round and yellow, 3 were wrinkled yellow, 3 were round and green, and the last one was wrinkled and green. Based upon such observations in dihybrid crosses between the plants differing in two traits, Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair. Means going of R in the gamete is not dependent upon Y, and vice versa. Means going of Y allele in the gamete is not dependent upon R. Right? So this probability table can be read from the book only. Otherwise, I will be explaining the diagram. 4.3.2 Chromosomal Theory of Inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons. It remained unrecognized till 1900. Firstly, communication was not easy as it is now in those days, and his work could not be widely publicized. Secondly, his concept of genes or factors in Mendel words are stable and discrete units that controlled the expression of traits, and of the pair of alleles which did not blend with each other, was not accepted by his contemporaries as an explanation of the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors or genes are discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900, three scientists, De Vries, Correns, and von Schirmack, independently rediscovered Mendel's results on the inheritance of characters. Also, by this time, due to advancements in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appear to be double and divide just before each cell division. These were called chromosomes or color bodies as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosomes during mitosis, which is equational division, and during meiosis, which is reductional division. The important things to remember are that chromosomes as well as genes occur in pairs. The two alleles of a gene pair are located on homologous site on homologous chromosome. Table 4.3, one side chromosomes point are being given and on the other side gene points are being given to you. During anaphase of meiosis 1, the two chromosomes pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of four different colors in the left and right columns. In the left column, which is possibility 1, orange and green is segregating together. But in the right hand column, which is possibility 2, the orange chromosome is segregating with the red chromosome. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes will lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis of the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and the female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscopes as well. Now, whatever keywords you have grasped by the reading till now, you can move ahead and write it in the chat. We will soon start with the 4.3.3 linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow-bodied, white-bodied females to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other and the F2 ratio deviated very significantly from the 9:3:3:1 ratio expected when the two genes are independent. Morgan and his group knew that the genes were located on the X chromosomes and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. Morgan and his group also found that even when the genes were grouped on the same chromosome, some genes were very tightly linked while others were loosely linked. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wings showed 37.2% recombination. His student Alfred Sturtevant used the frequency of recombination between the gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, gene maps are extensively used as a start point in the sequencing of whole genomes and was done in the case of human genome sequencing project described later. Polygenic inheritance, which is topic 4.4. Mendel's studies mainly describe those traits that have distinct alternate forms such as flower color, which are either purple or white, but if you look around you, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we do not just have tall or short people or two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by three or more genes and are thus called polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. The effect of each allele is additive. To understand this better, let us assume that three genes capital A, capital B, capital C control skin color in human with the dominant forms. Capital A, capital B and C are responsible for dark skin color and the recessive forms small a, small b and small c for light color skin. The genotype with all the dominant alleles will have the darkest skin color and that with the recessive alleles will have the lightest skin color. As expected, the genotype with 3 dominant and 3 recessive alleles will have an intermediate skin color. In this manner, the number of each type of the alleles in the genotype would determine the darkness or lightness of the skin in an individual. 4.5 Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Such a gene is known as a pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. An example of this is a disease phenylketonuria which occurs in humans. The disease is caused by the mutation in the gene that codes for the enzyme phenylalanine hydroxylase. It is a single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. 4.6 Sex determination. The mechanism of sex determination has always been a puzzle before the geneticist. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetic chromosomal basis of sex determination. Henking in 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects. And it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Henking gave a name of this structure as the X body, but he could not explain its reason or significance. Further investigations by other scientists led to the conclusion that the X body of Henking was in fact a chromosome, and that is why it was given the name X chromosome. It was observed that in a large number of insects, the mechanism of sex determination is the exotype, that is, all eggs bear an additional X chromosome besides the other chromosomes, autosomes. On the other hand, some of the sperms bear the X chromosome where some do not. Eggs fertilized by sperm having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome becomes the male. Do you think the number of chromosomes in the male and female are equal? Due to the involvement of the X chromosome in the determination of sex, it was designated to be the X chromosome, and the rest of the chromosomes were named as autosomes. Grasshopper is an example of exotype of sex determination in which the males have only one X chromosome besides the autosomes, whereas the females have a pair of X chromosome. These observations led to the investigation of a number of species to understand the mechanism of sex determination. In a number of other insects and mammals, including man, XY type of sex determination is seen where both male and female have same number of chromosomes. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called Y chromosome. Females, however, have a pair of X chromosome. Both males and females bear same number of autosomes. Hence, the males have autosomes plus XY, whereas females will have autosomes plus XX. In human beings and in Drosophila melanogaster, the males have one X and one Y chromosome, whereas females, they have a pair of X chromosome beside autosomes. Means the sex determination of human beings and Drosophila is same. In the above description, you have studied about two types of sex determining mechanisms, XO and XY type, but in both cases, males produce two different types of gametes, either with or without X chromosome, some gametes with X chromosome and some with Y chromosome. Such type of sex determination mechanism is designated to be the example of male heterogamety. In other organisms, example birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both males and females, but two different types of gametes in terms of the sex chromosomes are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird have been designated to be the Z and Y chromosomes. In these organisms, the female have one Z or Z and one W chromosome, whereas males have a pair of Z or Z chromosomes beside the autosomes. So next topic is 4.6.1 sex determination in humans. It has already been mentioned that the sex determining mechanism in case of humans is XY type. Out of 23 pair of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosomes are present in the female, whereas the presence of an X and Y chromosomes are determinant of the male characteristic. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry the X chromosome and the rest 50% has Y chromosome besides the autosome. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with the sperm carrying X chromosome, the zygote develops into a female or XX. And the fertilization of ovum with the Y chromosome carrying sperm resulting into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident in each pregnancy there is always a 50% probability of either a male or a female child. It is unfortunate that in our society, women are blamed for giving birth to female children and have been ostracized and ill treated because of this false notion. Parthenogenesis will be read under the topic 4.6.2 sex determination in honeybee, right? The sex determination in honeybee is based on the number of sets of chromosomes an individual receives. An offspring formed from the union of a sperm and an egg develops as a female, queen or worker, and an unfertilized egg develops as a male or drone by means of parthenogenesis. This means that the male have half the number of chromosomes than that of the female. The females are diploid having 32 chromosomes and males are haploid having 16 chromosomes. This is called haplodiploid sex determination system and has a special characteristic features such as the males produce sperms by mitosis and they do not have father and thus cannot have sons but have a grandfather and can have grandsons. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? All the students who are watching to this video, who are hearing to this live, you must have grasped few terms by the reading of this chapter. Type any 3 terms that you are more comfortable in now than in the beginning of the chapter. Are you interested in having the diagram description of each chapter? If yes, do comment me in the chat. I will definitely make a summary of all the descriptives, all the descriptions of the diagram given in a particular chapter. Or do you want it in a short form? What is more easy to you? Please let me know. Next topic 4.7 mutation. At the rate crows01. Hi, thank you for joining this live. I have already explained this. The video is already there in my playlist. You can go to the playlist of this chapter, Principles of Inheritance and Variation, and you will easily be able to understand. This is just a reading video. I'll get back with more explanatory videos once my live of reading will be done. Mutation is a phenomenon which results in alteration of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. As you will learn in chapter 5, one DNA helix runs continuously from one end to the other in each chromatid. It is highly supercoiled form, therefore loss or deletions or gain due to insertion or duplication of a segment of DNA result in alteration of chromosomes. Since genes are known to be located in chromosomes, alteration in chromosomes results in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancerous cells. In addition to the above, mutation also arises due to change in a single pair of DNA. This is also known as point mutation. A classical example of such a mutation is sickle cell anemia. Deletions and insertions of base pairs of DNA causes frame shift mutations. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutations. These are referred to as mutagens. UV radiations can cause mutations in organisms and it is a mutagen. So whichever topics I have read till now, you can write the keywords that you are more comfortable with now. 4.8 Genetic disorders. Pedigree analysis. The idea that disorders are inherited has been prevailing in human society since long. This was based on the heritability of certain characteristic features in families. After the discovery of Mendel's work, the practice of analyzing inheritance pattern of traits in human beings began. Since it is evident that control crosses that can be performed in pea plants or some other organisms are not possible in case of human beings, study of the family history about inheritance of a particular trait provides an alternative. Such an analysis of traits in a several of generations of a family is called pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure 4.13. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. DNA is a carrier of genetic information. It is hence transmitted from one generation to the other without any change or alteration. However, changes or alteration do takes place occasionally. Such an alteration or change in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. Mendelian disorders. Broadly, genetic disorders may be grouped into two categories, Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by alteration or mutation in a single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principles of inheritance. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia. It is evident that the X-linked recessive trait shows transmission from carrier female to the male progeny. A representative pedigree is shown in figure 4.14 for dominant and recessive traits. Discuss with your teacher and design pedigree for characters linked to both autosomes and sex chromosome. Color blindness. It is a sex-linked recessive disorder due to defect in either red or green cone of eye resulting in failure to discriminate between the red and the green color. This defect is due to mutation in certain genes present in the X chromosome. It occurs in about 8% of males and only about 0.4% of the females. It is because the genes that led to red-green color blindness are on the X chromosome. Males have only one X chromosome and female have two. The son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. Hemophilia. This sex-linked recessive disease which shows its transmission from an unaffected carrier female to some of the male progeny has been widely studied. In this disease, a single protein that is a part of the cascade of proteins involved in the clotting of the blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female, which is a carrier for hemophilia, may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because mother of such female has to be at least carrier and the father should be hemophilic. Unviable in the later stage of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Sickle cell anemia. This is an autosome linked recessive trait that can be transmitted from parents to the offspring when both the partners are carriers for the gene or heterozygous. The disease is controlled by a single pair of allele HBA HBS. Out of the three possible genotypes, only homozygous individuals or HBS HBS show the disease phenotype. Heterozygous HBA HBS individuals appear apparently unaffected, but they are carrier of the disease and there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The effect is caused by the substitution of the glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin chain results due to the single base substitution at the 6th code of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of this in the brain results in mental retardation. These are also excreted through the urine because of its poor absorption by the kidney. Thalassemia. This is also an autosome-linked recessive blood disease transmitted from parents to the offspring when both the partners are unaffected carrier for the gene or heterozygous. The defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chains, alpha and beta chains that make up the globin. This causes the formation of abnormal hemoglobin molecules resulting into the anemia, which is characteristic of the disease. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. In alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of the beta chain is affected. Alpha thalassemia is controlled by two closely linked genes, HBA1 and HBA2, on chromosome 16 of each parent, and it is observed due to the mutation or deletion of one or more of the two to 4 genes. The more genes affected, the less alpha globins are produced. While beta thalassemia is controlled by a single gene, HBB, on chromosome 11 of each parent and occurs due to mutation of one or both the genes. Thalassemia differs from sickle cell anemia in that the former is quantitative problem of synthesizing too few globin molecules, while the later is qualitative problem of synthesizing an incorrectly functioning globin. Chromosomal disorders. Next topic. The chromosomal disorders, on the other hand, are caused due to the absence or excess or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes, which is known as aneuploidy. For example, in Down syndrome, results in the gain of extra copy of chromosome number 21. Similarly, Turner syndrome results due to the loss of a X chromosome in human females. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomena is called polyploidy. This condition is often seen in the plants. The total number of chromosomes in a normal human cell is 46 or 23 pairs. Out of these 23 pairs are autosomes and one pair of chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome respectively. Such a situation led to a very serious consequence in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common examples of chromosomal disorders. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, thus known as trisomy of 21. This disorder was first described by Langton Down in 1866. The affected individual is short-statured with small round head, furrowed tongue and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. Klinefelter syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into the karyotype of 47 XXY. Such an individual has overall masculine development. However, the feminine development or development of breast, which is also known as gynecomastia, is also expressed. Such individuals are sterile. So whatever you have grasped till now, please move ahead and write any 5 keywords related to mutations. The last topic, Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosome, that is 45th with XO. Such females are sterile as ovaries are rudimentary, beside other features including lack of other secondary sexual characters. So with this, we are done with the reading of principles of inheritance and variation. Now type the law that now feels unshakable to you. See you tomorrow with another live. Stay connected, and if it was helpful, do leave with a like button. Even you can subscribe. It will be helpful.